Excellent. All right. Well, it's a pleasure to be here. And today I thought I would focus on talking about collaborations within the microbiome sphere. There are other experts here on working with global communities, and I'm happy to kind of talk about that in the panel and the questions. But I wanted to focus on a set of issues that are very upfront for me. So a lot of my work involves comparative analyses of gut microbiomes from across the world, primarily in non-human primates, but sometimes also involving human populations, as Dr. Rafaela mentioned. And so I want to talk about kind of how these collaborations can be very beneficial, but also some of the kind of risks and potential negative consequences and kind of how I think about those. So I'll just throw out kind of some ideas, and then I'm happy to talk about them later in the broader discussion. So I want to talk a little bit about kind of the intellectual benefits and potential drawbacks of these collaborations, as well as the material benefits and drawbacks of these collaborations. So I'll start by talking about kind of the intellectual side. So this is an example of a paper I published a while back that was comparing the gut microbiomes of 18 species of wild primates. And it was a really interesting data set and gave us a lot of insight into how important different factors were in determining primate microbiomes, such as diet or phylogenetic relationships, et cetera. And so this was a really kind of fun paper to write, and it's created a foundation for a lot of questions moving forward. It was a really powerful analysis that opened up a lot of insight and opportunities. So that was clearly an intellectual benefit to generating this sort of data set. But generating this data set was not a simple thing. It involved collaborating with 15 researchers across 11 countries to get the samples from all of these non-human primates. And so that's in terms of just getting the samples themselves. That doesn't include any of the lab work or the data analysis, et cetera. And so there's a lot of considerations kind of on the ethics side and kind of credit side when we have this many researchers and this many field sites involved in generating data. One of the things that's always in my mind is who gets to be a co-author, right? A lot of times with these big microbiome data sets, a lot of the emphasis is put on generation of sequence data or the actual analysis of the data itself, right? The statistics, the bioinformatic pipelines, et cetera. But this work requires a lot of time and resources in the field, right? So to get these samples, this is me from back when I was doing my PhD, right? People are spending days and days and hours in the field following non-human primates around, making sure that they're identifying the individuals and collecting the correct samples, getting additional sorts of behavioral data as it's needed, getting environmental data. And so this is a huge amount of person time. You also need grants and money to support this sort of research, which is in addition to all of the grants and money we need for the microbiome research. For my projects, when I was working on howler monkeys, I was primarily the person in the field collecting the data and doing all of this. But a lot of these long-term field stations, there's an actual building that has to be run, and people have field teams that they employ to help them collect these samples, right? And so again, in this case, it's not just the one person who's associated with that field site that I'm collaborating with, it's their whole team of researchers that they're paying to work at the field site year-round, and that they need to get grants to support this field team and to support the actual infrastructure at the field site. Okay. And so I always think about this in terms of, like I said, who gets to be a co-author? And because there are a lot of people involved in these processes, and it's not just about me generating the microbiome data, it's about all the people that are doing the hard work to get the actual samples. And so for me on my papers, I always include the field collaborators as authors. Now I leave it up to the person who I'm collaborating with directly to let me know who in the actual field team may deserve to be a co-author. But that's a really important part for me, is trying to understand who gets to be on that paper, and that it's not just me in my lab generating the data. Which brings me to another point. Microbiome data has been, or the microbiome field has been kind of at the forefront of creating open access to data, right? And on the bottom right hand, you can see there's a lot of benefits. I stole this from an ERC website. there's a lot of benefits to having open access data, right? You're encouraging and facilitating more research. You're making research transparent, right? You're reducing the potential data loss if people kind of lose the data or it degrades if they don't have sufficient resources to kind of back up the data at their own institutions, right? And a lot of times these government grants or other sorts of publicly funded institutions would like the data to be shared, right? Because there's taxpayer dollars, et cetera, going into collecting them. This all makes a lot of sense. But I sometimes worry about what comes along with open access data in terms of everything that I just spoke about, right? There's a whole team of people behind the collection of those samples. And as soon as sequence data goes open access, anybody can use it and generate it, which on the one hand is great. On the other hand, there's no requirement for the people that collected the samples in the field to actually be part of subsequent papers, right? And sometimes that might make sense if there's, many studies and every single study is just a small point on a figure. But in other cases, if data sets are really kind of pushing the main theme of this new story forward, it would be nice to consider kind of who collected those samples and where the data came from. And that's kind of a really difficult balance to strike, but I think it's an important thing that we really need to be thinking about with open access data. As well as the fact that if the first time you're publishing something, you have to put everything open access, that means it's like your one shot to kind of get everything out of those data you collected. And that can sometimes be difficult as well, right? You may have other things you want to do with the data, but you're not quite there yet in your own team. But once things go online, everybody else has access to them. So this is something I kind of struggle with in my own mind as well. I guess the other point I'll put on there is that some of my collaborators in other countries, those countries actually have laws about them having to be a co-author on the papers that involve their samples and their data. These are types of laws and rules that are trying to work against the type of biopiracy that Alyssa and Shani were talking about. And again, once data goes open access, people can grab that data and they don't necessarily understand that there were rules around kind of, those data can be open, but somebody needs to be a co-author. I have some of the same issues with sample sharing. I have a really wonderful collection of samples from many different non-human primate species, as well as some human populations. And I often get people who are interested in working with me and getting access to those samples so they can answer their own questions. And I'm really excited about doing that because there are things that we simply can't do in our lab or other techniques and questions that other people bring, and I want to be able to share them. But again, I have to have these conversations about the fact that those samples aren't necessarily just mine, that were collected in collaboration with people across the globe, and that we need to speak with those people and understand kind of how they want the samples to be used before I just start handing over the samples with only my name on them, because it wouldn't be ethical for me to pretend that it was, just the sample set that I own and that I'm in charge of. With this in mind, in my lab, what we actually do when we collect samples is if people send them in to us from their field site, we actually have our own lab sample number that we give to it that actually includes the name of the species, the field site, and the initials of the investigator that sent us those samples. And those sample names get used in the sequencing as well. So both the samples and the sequence data always have that identifier, so we know exactly where they're coming from. So I want to touch a little bit on material benefits. This speaks a little bit more to the human research, honestly, but I think it's true kind of more broadly in terms of thinking about kind of, again, these ideas of potential biopiracy or kind of getting scientific information out of kind of a diversity of global environments. So I think it's really important to think about what sort of information the people that are living or working at the actual field site are going to get. This is an example of an agricultural study that was done in the US. And so, these questions are also relevant in our own countries, not just when we're working in kind of other global locations. What kind of information is going back to the community that's participating in the project? And do they actually kind of understand enough about the project to be able to use that information and understand it? I think this is a really important question. And again, something that particularly in some of these kind of larger international collaborations that I worry about, because because of the comparative nature, I can't always be at every field site. And so I don't want to be participating in this kind of biopiracy or getting data from communities that they don't understand and can't use. And so I have to rely really heavily on my collaborators and their relationships with local communities, as well as provide whatever materials that I can to try to kind of avoid this. I also think again about these resources, right? Who gets access to these samples? So Alyssa and Shani mentioned the microbiome ARC, and there are some variations of that project where they talk about ethics and talk about how kind of each country has a representative that's in charge of their contributions to the ARC, which I think is a nice effort towards trying to make sure that these resources are available to the communities that they're coming from, that they could access those samples, for example. Um, but I would also remind people that there are social hierarchies and dynamics that are happening kind of politically and socially, even within other countries, that mean that just putting one person in charge of that country's contributions to this sort of arc doesn't necessarily mean that you're safeguarding them, right? There still can potentially be issues of equity and who gets access to those samples. Um, and then innovation is also something I want to think about in terms of kind of who are the participants of the study, right? Are the studies being designed in a way that they're going to address questions that are actually relevant to that community and that the community could benefit from, right? Are we actually trying to develop, in some cases, kind of these health interventions or prevention measures? Are we using the right populations to try to generate those things? And if we do generate them, are those communities going to actually have access to them, right? A lot of the personalized medicine kind of goals that are involved in a lot of microbiome research moving forward are going to be things that are highly costly. And are these sorts of precision medicine tools things that can actually be utilized by the communities from which the data are being generated? I think right now, in a lot of cases, the answer is no. And so I think we need to think really carefully about how we can make sure that communities can get access to some of the benefits from this actual research. So I'll leave it at that for now. Like I said, I just wanted to throw some ideas out there, and I look forward to discussing this more with everybody else. Thanks so much.